Welcome to Biology, the scientific study of life. Segmentation pattern. The bichord protein and other proteins encoded by egg polarity genes regulate the expression of some of the embryo's own genes. Gradients of these proteins bring about regional differences in the expression of segmentation genes. The genes of the embryo whose products direct formation for segments after the embryo's major body axis are defined. In a cascade of gene activation, sequential activation of three sets of segmentation genes provides the positional information for increasingly fine details of the animal's modular body plan. The three sets are called the gap genes, the parallel genes, and the segment polarity genes. The products of many segmentation genes, like those of the egg polarity genes, are transcription factors that directly activate the next set of genes in the hierarchical scheme of pattern formation. Other segmentation genes operate more indirectly, supporting the functioning of the transcription factors in various ways. For example, some are components of cell signaling pathways, including signal molecules used in cell-cell communication and the membrane receptors that recognize them. Cell signaling molecules are critically important once plasma membranes have divided the embryo into separate cellular compartments. Working together, the products of egg polarity genes like bichord regulate the regional expression of gap genes, which control the localized expression of parallel genes, which in turn activate specific segment polarity genes in different parts of each segment. The boundary and axis of the segments are now set. In the hierarchy of gene activations responsible for pattern formation, the next genes to be expressed determine the specific anatomy of each segment along the length of the embryo. Identity of body parts in a normal fly structure, such as antennae, legs, and wings, develop on the appropriate segments. The anatomical identity of the segments is set by master regulatory genes called homeotic genes. These are the genes discovered by Edward Lewis. Once the segmentation genes have staked out the fly's segments, homeotic genes specify the types of appendages and other structures that each segment will form. Mutations in homeotic genes can cause an entire structure characteristic of a particular segment of the animal to arise in the wrong segment, as Louise observed in flies. Like many of the egg polarity and segmentation genes, homeotic genes encode specific transcription factors. These regulatory proteins are gene activators or repressors, controlling expression of genes responsible for particular anatomical structures. For example, a homeotic protein made in the cells of a particular head segment specific antenna development. In contrast, a homeotic protein active in a certain thoracic segment selectively activates genes that bring about, or activates genes that bring about leg development. A mutant version of the gene encoding the thoracic homeotic protein causes the protein to be expressed also in the head segment. There is overrides the normal antennal gene activating protein, labeling the segment as thoracic instead of head and causing legs to develop in place of antenna. Scientists are now busy identifying the genes activated by the homeotic proteins, the genes specifying the proteins that actually build the fly structures. The following follow chart summarizes the cascade of gene activity in the Drosophila embryo. Notice that hierarchy of gene activation or activity in area Drosophila development. Maternal effect genes, egg polarity genes, gap genes, parallel genes, segment polarity genes, homeotic genes of the embryo, other genes of the embryo, and these are segmentation genes of the embryo. Although this simplified summary suggests a strictly sequential series of gene activation or action, the reality is more complicated. For instance, the genes in each set not only activate the next set of genes but also maintain their own expression in most cases. Amazingly, many of the molecules and mechanisms that revealed by research on fly pattern formation have turned out to have a close counterparts through the animal kingdom. The homeotic genes and their products exhibit these similarities in a most striking way. C. elegans, the role of cell signaling. The development of multicellular organisms requires close communication between cells. Indeed, even before fertilization, Drosophila molecules made in neighboring nerve cells cause localization of bicode mRNA at one end of the egg, thus helping to establish the anterior end of the future embryo. Once the embryo is truly multicellular, with membranes enclosing each individual nucleus and accompanying cytoplasm and decor. Signaling among the embryos signaling among the embryo's all cells becomes increasingly important. As we have seen, the ultimate basis of the differences between cells is transcriptional regulation, the turning on and off of specific genes. It is induction, signaling from one group of cells to an adjacent group. That brings about differentiation in some cases. Cell signaling also leads to the programmed death of specific cells, a phenomenon that is also crucial to normal embryonic development. The nematode C. elegans has proved to be a very useful model organism for investigating the rules of cell signaling, induction, and programmed cell death in development. Researchers know the entire ancestry of every cell in the body of an adult C. elegans, the organism's complete cell lineage. This information can be represented in a cell lineage diagram, somewhat like a pedigree that shows the fates of all the cells in the developing embryo. Because the lineage of each C. elegans cell is so reproducible, scientists at first thought that it must be determined from the start, suggesting that cytoplasmic determinants are the most important means of establishing cell fate in nematodes. However, although cytoplasmic determinants do play a key role very early in C. elegans development, a combination of genetic, biochemical, and embryological approaches have revealed important contribution of inductive events as well. Induction as early as the fourth stage in C. elegans cell signaling helps direct daughter cells down the appropriate pathways. For instance, as shown this figure, notes that a signal from cell 4 acts on cell 3. Notes that closer look, posterior here, force signal, protein 4, and this one 3. So that one of the daughter cells of cell 3 eventually gives rise to the intestine. The signal is a cell surface protein made by cell 4 that can be recognized and bound by a cell surface receptor protein on cell 3. This interaction triggers events inside cell 3 that result in one end of the cell, the posterior end, becoming different from the other. When cell 3 divides, the posterior daughter cell will go on to make the intestine, whereas the anterior daughter cell has a different fate. If cell 4, notes that experimentally removed early in the 4th cell stage, no intestine forms, but if an isolated cell 3 and cell 4 are recombined, the intestine develops as normal. These results helped researchers recognize the role of induction in early nematode development. Induction is also critical later in nematode development as the embryo passes through three larval stages prior to becoming an adult. The vulva, the tiny opening through which a worm lays its eggs, arises a worm arises from 6 cells that are present on the ventral surface at the second larval stage. Notice this figure. Notice that also a single cell of the embryonic gonads, the anchor cell, initiates a cascade of inductive signals that establishes the phase of the six vulvar precursor cells. If an experimental destroys the anchor cell with a laser beam, the vulva fails to form the precursor cells, simply becomes part of the worm's epidermis. The signaling mechanisms in both of these examples are similar to those discussed in Chapter 11. Secreted growth factors or cell surface proteins bind to a receptor on the recipient cell, initiating intracellular signal transduction pathways, transcriptional regulation, and differential gene expression in the induced cell are the usual results. These two examples of induction during nematode development illustrate a number of important concepts that apply elsewhere in the development of C. elegans and many other animals. In the developing embryo, sequential inductions drive the formation of organs. The effect of an inducer can depend on its concentration, just as we saw with cytoplasmic determinants in Drosophila. Inducers produce their effects via signal transduction pathway similar to those operating in adult cells. The induced cell's response is often the activation or inactivation of genes, transcriptional regulation, which in turn establishes the pattern of gene activities or activity characteristic of a particular kind of differentiated cell. Okay, thanks for listening.